Most animal and plant cells are about 0.01 to 0.1 millimeters in length. On your screen now, there is an example of an animal cell. This is a cheek cell. And an example of a plant cell. This is a leaf cell. These two cells are about the same size, but plant cells can grow much larger than animal cells. For the exam, you don't need to memorize cell lengths such as 0.07 millimeters, but you do need to demonstrate an understanding of typical cell size, so you should know that cells don't typically exceed 0.1 millimeters. So how do eukaryotic cells, such as these, compare to prokaryotic cells, such as bacteria? Most bacterial cells are only about 0.001 to 0.01 millimeters in length. So we have our animal cell and our plant cell, and on the left is our bacterium. Now, if we were to actually compare the bacterium to the real-life sizes of animal and plant cells, it would actually look something like this. In fact, bacteria are around 10 times smaller than animal and plant cells. We can see a few of the largest animal cells like egg cells with the naked eye, but we can't see bacteria without the help of microscopes. So then for the exam, you need to know that bacteria are much smaller than plant and animal cells, and you should memorize that they're typically about 10 times smaller. However, bacteria can form visible colonies when there are a large group of cells in the same place. In this diagram, you can see a Petri dish. These orange blobs on the Petri dish show bacterial growth. These orange blobs are the visible colonies made up of loads and loads of bacterial cells. What units do scientists use to describe and compare the size of cells? Scientists use different units to refer to smaller and smaller scales. Scientists use the prefixes centi, milli, micro, and nano. So let's use this table to compare these units to each other. Let's start with centimetres. So 1 centimetre is 1/100th of a metre. A millimetre is 1/1000th of a metre. A micrometre is 1/1000th of a millimetre. And a nanometre is 1/1000th of a micrometre. Let's put all of these units as a division of a metre in the bottom row. So a centimetre is 1/100th of a metre. A millimetre is 1/1000th of a metre. A micrometre is 1/1 millionth of a metre. And a nanometre is 1/1 billionth of a metre. So this means that 100 centimetres make up 1 metre, 1000 millimetres make up 1 metre, 1,000,000 micrometres make up a metre, and 1 billion nanometres make up a metre. For the exam, you need to know the relationship between all of these units. How do we convert between units? To convert between units, we multiply or divide by a factor of 10. So if we wanted to find the value 0.00001 centimetres in millimetres, we multiplied this value by 10. If we want to then convert from millimetres to micrometres, we multiply by 1000. And to get from micrometres to nanometres, we multiply by 1000 again. You can see how the decimal place has moved between centimetres going all the way down to nanometres. So when we convert to a smaller unit, we move the decimal point to the right. If we wanted to go back from nanometre to micrometre, we will have to divide by 1000. To convert from a micrometre to a millimetre, we divide by 1000 again. And to get back to centimetre from millimetre, we divide by 10. By doing this, we're moving the decimal point over to the left again. You should always do a sense check when you're multiplying or dividing and question whether the answer you expect will be larger or smaller than the value you start with. Now, if you wanted to go from a millimetre straight to a nanometre, instead of multiplying by 1000 twice, we can just do one step and multiply by 1,000,000. This is the same as doing 1000 times 1000. So now you know how to convert between units, but how do we know which units to use? Scientists measure most cells in micrometers. Remember, when you're converting from millimeters to micrometers, you multiply your value by 1000. So compared to the millimeter scale at the top, the micrometer scale will look like this. So 20 is 1000 times larger than 0.02, 40 is 1000 times larger than 0.04, and so on down the scale. So if we were to place animal, plant, and bacterial cells on this scale, this is the range that the animal and plant cells would fall into. Bacterial cells will fall all the way down on the far left-hand side of the scale. Remember, these are smaller than animal and plant cells. So we measure whole cells in micrometers, but how do we measure smaller subcellular structures? For small subcellular structures such as ribosomes, scientists often use the nanometer. Remember, when you convert micrometers to nanometers, you also multiply by 1000. So our nanometer scale will look like this compared to micrometers and millimeters above. So we're moving down the scale in terms of how large these measurements are, with millimeter being the largest and nanometer being the smallest. This is where ribosomes will fit on this scale. The maximum size of ribosomes in eukaryotes is about 30 nanometers, which is easier to say than 0.00003 millimeters.
Thanks for watching. If you want to take your GCSE revision to the next level, head over to launchpadlearning.com and check out our smart learning platform that's been designed to get you top results in your exams. We cover your whole specification and make revision fun with interactive quizzes, easy-to-follow videos, and more. You'll be kept motivated by your own AI tutor who's here to support you every step of the way. To check it out for yourself, click here. Or click here to keep watching a selection of the videos from our full GCSE Biology course. See you there!